Now we'll take the opportunity to work through some diffusion and osmosis problems looking at semi-permeable membranes and the flow of materials into and out of a cell. Here's our first example problem. You can see the materials that typically make up a cell. There's water, proteins, lipids, inorganic ions, sodium, potassium, calcium, magnesium, etc., nucleic acids, carbohydrates, which include sugars and starches, and just some other substances in there as well. Now what will typically happen is we've got to figure out which materials are able to permeate through the membrane. And in this case it's water, inorganic ions, sugar, and the other substances. So that would mean that lipids are unable to flow, and nucleic acids are unable to flow, and starch is unable to flow, and proteins are unable to flow as well. So if this is the case, then we'll look at water first. Water is of higher concentration on this side, so by osmosis it will go through to the other side from high to low concentration. If we start looking at the materials in here, the inorganic ions are higher in concentration on the outside, so some of those will seep into the cell through the semi-permeable membrane going from high to low. Nucleic acids are unable to move as well, and as we look at our carbohydrates, sugar is a little bit more concentrated on the outside, so it will flow into the cell from high to low. And other substances are more concentrated on the inside, and they will flow out of the cell from high to low. So all in all, water will flow out, other substances will flow out, and sugar and our ions will flow into the cell. The other things will not flow at all because they cannot go through the membrane because it is only semi-permeable. It does not let everything in and out. Here's our next problem. Once again, we can forget about proteins right away, lipids right away because they can't go through, nucleic acids we can forget about right away, starches right away. Those things are too big to flow through the semi-permeable membrane. So we'll start with water. The higher concentration is here, so through osmosis, water will go into the cell from high to low. And now we start looking at diffusion and the other different substances. Now as you look at the inorganic ions, notice it is equal on both sides. That does not mean that it will not flow. It means that it will go in and out at the same rate. This is called dynamic equilibrium. Dynamic means moving, and equilibrium means balance. So things will flow into and out of the cell at the same rates, maintaining both those concentrations. As we look at carbohydrates here, sugars will flow into the cell from high to low, and other substances will flow into the cell from high to low as well. And so in summary, if you're looking at this, there is a lot of stuff flowing into the cell. Remember, these inorganic things go back and forth, but including the water. And so this cell might actually expand and burst, and that would be a problem. But as you look at it, and you might go, why do we even bother looking at the concentrations of proteins or lipids or nucleic acids or starches? Because of the concentrations of those are so small on this side, it does have some effect on the flow of things into and out of the cell, because everything else tends to be forced into the cell. So even though proteins, lipids, nucleic acids, etc. cannot move into the cell, they still do have an effect on what can move into and out of a cell. Here's our third problem. Once again, I'm just going to scratch out the proteins, lipids for starters. Nucleic acids, don't need to worry about those, don't need to worry about starches. We'll start with water. Osmosis says from high to low. So some of the water will leave the cell through osmosis. As we look at diffusion now, inorganic ions go in from high to low, sugars go in from high to low, and this time our other substances have our dynamic equilibrium, flowing in and out at the same rate. Here's our last problem. Once again, the proteins, lipids, those are not going to flow through. But I do want to point out, if you look at the nucleic acids and the starches, they are equal on both sides, but they're not going to move through it because it's a semi-permeable membrane and they are too big. So in this case we have an equilibrium like we had in some of the other problems, only it's not exactly the same. It's what we would call static equilibrium because it's not moving, it's not dynamic. If we look at water, water's the same on both sides, so that will be dynamic equilibrium because it can flow through from side to side, and that's osmosis once again. And as we start looking at diffusion, we have dynamic equilibrium, between the inorganic ions. They'll flow back and forth. Our sugars go from high to low, which is into the cell to out of the cell, and our other substances will flow out of the cell as well. So our net flow of things is going to be out of the cell, which means the cell is going to shrink or shrivel a little bit. Sometimes, as we look at these different solutions, is that if the outside of the cell is hypertonic, that means high concentration, water will tend to flow out to try to balance and reach that equilibrium. And we have plasmolized the cell. It will shrivel up and will not do very well. If the flow is dynamic equilibrium here, or the same amount of water is flowing in and out, the cell tends to be flaccid or just a little bit loose. If more water is going in because the outside is hypotonic and the inside is hypertonic, then the cell becomes turgid and firm. This is why you might soak celery in water when it's in the refrigerator, so that the celery stays nice and firm and doesn't shrivel up and become flaccid. So these are some of the things that go on if too much water goes into a cell and there's too much trigger pressure, it can break, which is called lysis. And obviously if too much water leaves the cell over here, the cell can die as well.